We're going to be talking about AI in transforming science, everything from energy storage to biotechnology. We have Qichao Hu from the SES AI Corporation and James Collins moderating the conversation. Good luck.
Great. Well, I would like to welcome you all to this afternoon's session. We're just going to start off with some quick intros. So I'm Jim Collins. I'm actually on faculty at MIT. I'm a biological engineering professor and the Termeer professor of medical engineering, and I also serve as a faculty co-lead of our Jamil Clinic, which is our nucleating point for AI for healthcare at MIT. Beyond MIT, I'm also on faculty at the Broad Institute of MIT and Harvard, which focuses largely on genome efforts, as well as the Wyss Institute at Harvard, which is biological engineering. And I'll be moderating, but in fact participating in a conversation with Qichao this afternoon. And just briefly, our lab's been focusing on AI for science, specifically around using it to discover and design new antibiotics. And our motivation is really quite simple, is that we're actually facing a healthcare crisis that's dramatically underaddressed. And that's around antibiotic resistance. So resistance has been growing decade upon decade. These nasty pathogens have in the past been largely limited to hospitals. Worst place to be when you're sick is a hospital because of these superbugs, but now they're out in our communities. They're in our childcare centers, athletic fields, shopping centers, et cetera. And unfortunately, this is coupled with an anemic pipeline. Biotech and pharma are getting out of antibiotics. It doesn't make sense for them to do it. It costs just as much to develop an antibiotic as it does a cancer drug or a blood pressure drug. But an antibiotic you might take once or maybe over 3 or 4 days. Only costs you a few bucks, maybe $100. Cancer drug, blood pressure drug, you take for many months, years, if not the rest of your life, and you could be charged many thousands, hundreds of thousands of dollars. The UK Commission's actually estimated if we don't face this crisis, we could have up to 10 million deaths per year from antibiotic-resistant infections, so outpacing drugs from cancer by the year 2050. So at MIT, we actually took up this challenge of AI for good, AI for science, starting a few years ago to see could we use AI basically to kind of in a battle of our wits against the battle of the genes of these superbugs. And I'll share some more of that through this afternoon's discussion. So Qichao.
Yeah, thanks, Jim, for participating in this chat. And also, thank you for coming. I'm Qichao Hu. I'm the founder and CEO of SES AI. And then I come from the battery industry. And then we actually use AI to develop our batteries. And in fact, we actually use 3 types of AIs: AI for science, for the material discovery, AI for manufacturing to ensure quality, and then AI for safety. And then so to put things into perspective, the battery industry has been around for 30 years, but then the rate of innovation in the past has been very, very slow. And it takes about 10 years to develop a new battery chemistry and then commercialize this. So what we're trying to do is to use AI to screen a large universe of molecules so that we can speed up the material discovery for batteries. One example is in the last 30 years, the entire global battery industry only studied about 700 molecules. But there is 10 to the 11th molecules that could be used. So 100 billion, right? And then our goal is to study 100 billion in a year when the industry studied 700 in 30 years. This is the use of AI for science. And then also, from a manufacturing perspective, globally, this year, 2024, there's about 17 million EV cars sold. So 1 out of every 5 new cars sold is an EV. And there are roughly 1,000 batteries per car. So that's 17 billion battery cells. 17 billion battery cells. And from a quality perspective, roughly a good manufacturer can do parts per million. So 17 billion, that's 17,000 batteries that will catch on fire. So to prevent the EV fires, and that's why we're seeing a lot of fires today, we have to use new tools like AI to really address this. So Jim and I, and then we've been collaborating on using AI for science for more than a year, because the audience might think, well, what does antibiotics have to do with batteries? It's like 2 separate fields, but actually we've been collaborating for more than a year now to use actually quite similar tools to accelerate material discovery, both for antibiotics and also for batteries. So maybe Jim, if you could actually go deeper about what you do, and then how you use AI, and then how AI has helped accelerate your work.
Yeah, happy to do so. And I should say, we're going to have questions and answers toward the ends of this, but we can make it a living classroom. So if you have questions, just raise your hand. Like we are in an MIT classroom where we encourage our students to ask questions as we're going on. We don't need to just have it as a two-person dialogue. So give you just a sense of what we've been doing and how we started. So it goes back about 5 years ago. And we'll maybe speak to this a bit. I guess it's now almost 6 years ago, is that MIT recognized that we were asleep at the wheel for this third wave of AI. So the first wave, 50s and 60s, MIT really led it with folks like Marvin Minsky and Seymour Papert. And the second wave of AI in the 80s, where folks are focused on expert systems and language-based software like Lisp, MIT also played a leading role with folks like Patrick Henry Winston. This third wave that was focused on really emerging deep learning and large data sets, starting in the 2010s, MIT kind of realized that we weren't playing a leadership role. And so in the spring of 2018, MIT launched a campus-wide initiative to try to elevate AI as a central theme for around the Institute. And to launch it, we actually had a symposium to invite faculty who are doing AI or AI-adjacent work. And I was invited and actually met leading faculty at MIT, Regina Barzilay, who's been using AI in the context of a number of different medical challenges, including looking at chemical compounds. And we realized we had overlapping interest and decided to launch a collaboration to see if we could use AI to discover new antibiotics. Came out of, literally, we sat next to each other at this meeting, and what we also recognized, and many can appreciate, is that the golden age of antibiotics was in the 1940s, the 1950s, and the 1960s. So before I was born, before probably most in this room were born. And it preceded the revolution in AI, preceded the revolution in genomics, biotech, molecular biology, et cetera. Folks were basically finding bacterium in soil samples and then looking to see what they were producing, and from that actually pulled out most of the antibiotics that we use today. Regina and I thought, could we do something with AI? So we actually bootlegged a project. We were unfunded. And we looked around to see what did we have that we could use to begin to train models. And we pulled together a very small library of compounds. We did not want to have it small, but all we had was available with 2,500 compounds. So drugs. This included 17 FDA-approved drugs and 800 natural compounds. We applied these to a bacterial pathogen, E. coli, which many of us have heard. You hear about it in food outbreaks. It's actually involved in urinary tract infections as well. And interesting, it's a model organism used in many labs at MIT. We applied each of those 2,500 compounds to E. coli and asked which of them exhibit antibacterial activity as evidenced by growth inhibition. If the compound exhibited at least 80% growth inhibition, we said it was antibacterial and assigned it a 1. If the compound didn't achieve at least 80% growth inhibition, we said it wasn't antibacterial and assigned it a 0. We then took those featured data along with the structural information on each compound and began to train a deep graphical neural network. These are a form of an AI model that can actually look at graphical structures, in this case, structures of chemical compounds, kind of bond by bond. So think back to your chemistry classes, inorganic or organic chemistry, where you'll have a chemical structure, carbon and nitrogen and oxygen with lines between it. These models can look at those sets of atoms and lines and associate it with a feature, in this case, antibacterial or not, and then begin to build out each of those structures, substructure scaffolds, and begin to learn what compound structures might make for a good antibiotic. You can then feed it a new structure after you trained it and ask, is this a good antibiotic? And it will look and calculate from what it had learned about each compound bond and substructure, make a calculation between a 0 and 1. Could it make for a good antibiotic? We trained this model, then we applied again. Look, what did we have? And at the Broad Institute, we had a drug repurposing library of 6,100 compounds. And we applied the model to that and asked which is predicted to make for a good antibiotic and which doesn't look like existing antibiotics. So what's happened in the last 3 decades is we've rediscovered all of our favorite antibiotics again and again and again. Quinolones, penicillins, aminoglycosides. But when we asked which of these molecules out of 6,100 looked like a powerful antibiotic but didn't look like existing one, only one fit those criteria. And it's a compound we named Halicin. This was an homage to HAL, the killing AI computer system from 2001 Space Odyssey. They killed humans. We want this thing to kill bacteria. Anyways, it turned out Halicin was remarkable at killing bacteria. it killed multi-drug resistant, extensively drug resistant, and PAM resistant, so bacteria that are resistant to every known antibiotic, including TB, Clostridium difficile, Acinetobacter baumannii, and got people really quite excited, acted by a new mechanism of action. And on the basis of that, we were able to launch what we're calling the Antibiotics AI project at MIT that was funded by efforts and support from the Audacious Project, which is a new philanthropic effort run by TED, the group that does the TED Talks. And we now have also coupled with a group called Fair Bio, a nonprofit we launched, whose mission is to take the most promising molecules out of the Antibiotics AI project and move them towards the clinic through partnerships with a few remaining biotech and pharma still interested in this space, as well as other philanthropies, and notably Fair Bio. And our lab just got a great support from ARPA-H, a new innovative effort in the federal government to actually discover or design 15 new antibiotics through preclinical stage, really to try to address this growing health crisis. So maybe I'll pause there and pass it to you, Qichao, on what is happening in the bacteria battery space that is in part motivated by some of this AI effort.
Yeah, so in battery, you have four materials really: cathode, anode, electrolyte, and separator. And then one material that actually is key to a lot of the performance and then safety of batteries is the electrolyte. But then it's been really hard and really slow to develop electrolyte materials. And then I mentioned this earlier, the last 30 years, the entire global industry only studied about 700 molecules. So if you keep reformulating these 700 molecules, you can't really do much. So you're stuck with a battery chemistry that's outdated. So you can use that battery chemistry for EVs, but then you really can't use that for flying cars, for example, urban air mobility, and then robots and robotaxis. So, and then one limitation that we felt is really with human scientists. And then last year, on average, every week, about 700 to 800 papers were published every week, just in the battery space. And it's just impossible for human scientists to read and comprehend and understand that much information. So what we set out to do about 2 years ago is, okay, let's take all the possible small molecules that could be used for electrolytes in the entire universe, 10 to the 11th. And then we actually build this database. But then to train AI model, you really have to have data. And then so in our case, well, in the case of ChatGPT, it's really basically all the data the human race created in the last 200,000 years, all the books, all the knowledge, like social media, everything. So what is the equivalent data for molecules that we can train this AI? Well, that's basically all the fundamental physical properties, chemical properties of 10 to the 11th molecules. But this database actually does not exist. So we set out to build this database. Actually, we built probably the world's largest physical chemical properties of small molecules. So that includes like all the hormonal, the energy levels, the solubility, so at the single molecule level, at the solution level, and then at the interface level. So this takes a lot of computation. It's just computation chemistry. So you need compute power, and then you need really smart algorithms. And then so it turned out that NVIDIA's H100 is on average about 4x faster than the older A100, and then about 100 times faster than the CPU to do this kind of fundamental molecular level computation. So that means if you had done this in the 90s, it would have taken you probably 1,000 years, like 700 some years. But now we can do that in 10 months. And then we announced this partnership with NVIDIA, and then we have about close to 1,000 of the H100s, all dedicated to AI for science for battery electrolyte. So now we're going to map all the basic properties of all these small molecules. And then, like Jim mentioned, okay, and then the human scientists have a good understanding, okay, these properties tend to have good performance at low temperature, for example. These other properties tend to have good performance at high voltage. So we do have a good understanding the human scientists, and then we're also trying to systematically train this model with all papers, several million papers, and all books, several hundred books related to the battery field. And then all the internal meetings that we have, we record all the meetings, internal meetings, and then also lots of MIT open course videos on chemistry, material science. So we train all these models with all this information, external and internal, and then we map that onto this large database of physical chemical properties of molecules. And then recently we actually found a molecule that we use as a solvent in our lithium metal cells, and that demonstrated a 20% improvement in cycle life. So that's really exciting. And then lithium metal, it's one of the holy grails in the battery industry. It's really lightweight. So we've proven that this AI for science approach works. And then we actually found a molecule that delivered 20% improvement in cycle life for lithium metal. Now we're going to apply this tool for basically every lithium, sodium battery out there for different applications. So this part is really interesting. And I think the key thing is really two things. One is it takes the luck out of science discovery. I think in the past, in the battery field, like, of course we have lots of scientists, and then we do have a systematic human approach, but then sometimes you have to get lucky. So this takes the luck out of it, because basically you map everything. And the second is, the concept is not new. People had this concept in the 90s, just didn't have the H100s. So didn't have the compute power. So now that you take the luck out of it and you have the compute power to compute all the basic properties, I think what we've demonstrated with AI for science for lithium metal, and then it's been validated in one type of battery chemistry, I think we can apply this to a much broader field.
I should make a comment that the field I'm in on antibiotics is actually probably the best example of luck in science. And we're nearing the 100th anniversary of that lucky event, which was when Alexander Fleming discovered penicillin. So the backstory on that is that he had left a plate of bacteria out in his lab. He went on vacation, left the window open, and a fungal spore came into the window and landed on his plate and began to produce penicillin that actually inhibited growth of the bacteria where it grew. He came back, saw this contaminated plate, and serendipitously actually had the insight to see, oh, this contamination was making something that was actually inhibiting growth of the bacterium and actually could make for something interesting, and then was able to isolate and identify what was being made was penicillin. So I'm hopeful that AI will significantly enable us to minimize the need for luck and go. And to give you a sense of that, what happens now in the modern age for antibiotics is that we typically will do drug screening. So you either will use robots, so a liquid handling robot, or a human robot, an MIT graduate student, to introduce those compounds onto a bacterium and screen them. So it could be anywhere from the 2,500 that we use to, say, 1 million for a large pharma library. These things can be quite expensive, quite difficult to curate that many compounds. But to Qichao's point, AI now really allows us to relatively rapidly, with a trained model, to screen enormous libraries that in the past were not possible. So interestingly, most of now the in silico libraries are of order 10 to the 10. So the larger ones, the largest in my space, are about 10 to 30 billion. But in fact, to give you a sense of where I think we're only beginning to scratch the surface of the great diversity in chemical space, is that it's been estimated there are 10 to the 60 molecules that can make for good drugs. So again, for the MIT-related folks in the audience, you can imagine, we really are not even beginning to sample that enormous space. And I do think as we expand compute, it's really where AI is going to make a big difference, both in drug discovery and in the material space, and in particular in the energy space for batteries.
And then Jim, in the antibiotics, how do you know it works? Like, what's a roadmap from, say, developing a new material or having the AI recommending a new material and then proving that this actually works? What's the timeline and roadmap?
Well, the roadmap is really, it mirrors what happens in other aspects of biotech and pharma. So you'll come up with your molecule, your potential hit. in this case, initially, computationally, then you'll actually try it out in the lab in a dish. In my case, it's easy. You can actually drop it on bacterium, both those that are potentially susceptible to an antibiotic and also those resistant. If you actually kill or inhibit that growth, you then move to animal models, typically small animal models, mice, and then larger animals that could be typically dogs, and then you move into human trials. What's interesting in my world of antibiotics is I'm sure many have heard the old adage that we've cured cancer 900 times in mice and rats, but very few times in humans. And unfortunately, it's an adage that is true for the most part. In antibiotics, if you can kill the infection in a mouse, you have a 90% success rate of actually killing that infection in a human. So it's a marvelous space to invest in if you can get there. At present, our AI models are absolutely brilliant right now at predicting, discovering, and designing molecules that kill bugs in a dish. We're not very good at killing yet the bugs in the mouse. So again, given that you're here, you can appreciate, AI is at a crossroads in so many fields, including in medicine and biology, and we still lack a lot of data. So we don't have a lot of data on what makes for a good drug or a compound. Solubility, bioavailability, metabolic liability, is it toxic? We've now collected a lot of data. We've expanded our 2,500 compound library now to 39,000 at MIT, and we've used it against 7 different bacterial pathogens, including TB. And we've now applied it to 3 different human cell lines for toxicology. But we don't yet have those data that could really make it a success and make it easier. So for those of you interested in the finance world or biotech, that I think AI is going to increasingly become a valuable tool, a complement to kind of build on the previous discussion we just heard in this room. But there's a long way to go. And we need to get after much larger data sets in order to really harness the power of these tools.
Yeah, and then in the battery industry, for example, when we do AI for science, a lot of the data are synthesized. So we can just get GPUs and then compute. But then when we get to manufacturing, then a lot of the companies are in silos. It's hard to get access to that data. What about in your field? How do you get the data?
Yeah, it's again unfortunate that I think biotech, and pharma, they have significant profit motives, and they've committed a lot of resources to generate their in-house data and create their in-house libraries, which I think they tend to overvalue the proprietary value. But for the most part, these data and these resources are siloed, and with the exception of sequencing and I would say protein structure data, that in biology and medicine we're not yet in the big data phase. And so to speak, from our collaboration, where you're making really great progress, both discovery and design. The challenge is, can you synthesize the molecule? And the same holds in antibiotics. So I mentioned our discovery effort. We've actually just finished an effort where we've trained a number of models that are generative AI models to design new antibiotics. And we came up with hundreds of really cool-looking molecules. We shopped them around to 10 different CROs that could synthesize molecules, and they choked on our list. They basically came back and said, we couldn't do these. We then cut it down to about 40, and we got bites from 2 different groups. One is ChemScene, that's based here in the US and also has an outlet in China. Another called Etamine, notably Etamine, which is probably the go-to for small molecule synthesis in academic groups in biotech, is based in the Ukraine. And so they've been really quite busy for the last 2.5 years with a lot of other things happening in their country. But they've been the great group. And so each of these said, yeah, we can make each of these 40. And in the end, they only made 6. We got 6, but notably where there's excitement is that 5 of those 6 turned out to be potent new antibiotics. So we had in that case north of 70% success rate. And so our bottleneck really is, can you synthesize compounds? And I know in the battery space, it's also, can one come up with interesting new chemistries, and can you synthesize? And I do think AI is going to give us a better path to the synthesis routes, and can you get for the shortest path? And I think also point us to new and interesting chemistries in biology. But again, it's very early.
Yeah. Similarly, in the battery field, we can map 10 to the 11th molecules, but then our ability to synthesize, it's still like 100 years old. And then we make single-digit molecules per month. So the rate limiting is still in our ability to synthesize. We can map 100 billion and then screen to, say, hundreds of thousands, but then we can only make less than 100 per year because we only make single digit per month. So at the end of the day, the rate limiting, it's like AI would screen all the ideas and then recommend ideas, But then the humans are still the limiting factors in synthesizing the molecules and then formulating and then actually making the batteries and then testing that.
That's right.
Yeah.
That's right.
Yeah.
What do you see as some of the concerns, the potential societal concerns and or need for regulation in AI, say, as applied to the battery and material space?
Yeah, I think in terms of materials, the society cares more about safety, especially safety of electric cars. And then recently we see fires, just electric cars catching on fires around the world. And then people want to make sure that when we develop new materials, we have safety in mind. So you want to make sure you design safe materials and then not toxic materials. Some of the materials used in batteries, the salt and the additives, they may improve performance, but they're toxic. They are really bad for the environment, and you cannot recycle that. So ability to embed criteria in the AI models that you only recommend new molecules that are environmentally friendly and also not super expensive.
Yeah, it's interesting. In the antibiotic space, we came out with a piece where we showed you could use AI to discover a new antibiotic to kill Acinetobacter baumannii. So Acinetobacter baumannii, which is also known as the Iraqi bug, is a bug that's endemic to Iraq and Afghanistan. It actually really troubled U.S. and U.K. soldiers during the Iraq and Afghanistan war, where they would get skin wounds and they would get infected with Acinetobacter baumannii, and it's not a very susceptible bug to our existing antibiotics. It's actually at the top of the WHO's list of nasty bacterial pathogens for which we desperately need new antibiotics. A year and a half ago, we showed AI could come up with a new compound called abaucin that goes after, and why I bring it up is that this was just as nation states around the world were really getting worried about AI. ChatGPT had been out for a few months. Folks were worried about what could this be used for. Folks were worried about how, not ChatGPT, but AI could be used in autonomous weaponry, et cetera. And antibiotics was actually celebrated as a case of AI for good, and of a type not to regulate too much because you want to see this advance, which I found interesting. And then we got caught in the backwind on the other side of it, in that the end of last year, in December 2023, we published a piece in Nature on showing that you could use explainable AI to gain insight into the chemical structures that were being learned by an AI model to help you to identify a new class of compounds, new class of antibiotics, in this case to go against MRSA, probably the most famous antibiotic-resistant pathogen. And in the context of that, we also showed you could train models to develop toxicology models to identify compounds that were toxic against human cells, and we used this to identify molecules that were not toxic. That was our intent. Well, after we published, the guys in the black suits showed up in my office at MIT because they wanted to talk to me about our toxicology models and how the bad guys could use it. And I was intrigued because I hadn't thought of this. I mean, as many in this room can appreciate, and certainly those who train on our campus, that all technology can be dual use. And in this case, they were actually worried that these models could be used to discover really toxic compounds. And unfortunately, the answer is yes. It could be used to find new toxic compounds in nature and or to design new ones. And there are plenty out there, and they usually are the source for bioweaponry. And we actually have a number of countermeasures already for existing ones, toxins of various forms. But unfortunately, this could open up the possibility that you could find ones that could go around the countermeasures. We actually have support from the Department of Defense in our lab to actually use AI to come up with more countermeasures for tox molecules. But it's interesting of how this came back on largely an AI for good, but it's interesting that there are some bad guys out there you always have to keep in mind.
Yeah, and then so we put batteries in cars, and then we found the explainability of these AI for science models. For example, the model can recommend good molecules, and then it can show improvement in performance. But then if we're not able to explain the mechanism or not able to understand the mechanism, people don't really trust it. And I think if they can understand the mechanism, they will trust it a lot more. But then how these models will actually find new molecules that can improve performance, we also don't know. A lot of times we only know the basic properties, and then we try to avoid teaching the model with our understanding of mechanism, because our understanding of mechanism could be incorrect. So that's one thing. So we have found the model can come up with molecules that can improve performance, but then the mechanism I think we're behind in terms of understanding that. Coming back to, so this is the MIT AI conference, and then recently the Nobel Prize in Chemistry was awarded to Team DeepMind. How do you think MIT compares with its peers in this field?
MIT is an amazing, university, one of the top in the world. But if you compare us to some of the top groups in AI, I think we're still behind, unfortunately. So I think we're behind Stanford. I think we're behind University of Toronto. I think we're behind University of Cambridge. And interestingly, given you brought up Google DeepMind, I think that this area of AI, it's where, in fact, most of the best research is being done in industry right now and not in the universities. And the universities are doing some of the smaller scale blue sky work, as well as, of course, training the next generation. But I think MIT has a way to go. I think that we lack compute power. So I think folks in the room would be surprised at how limited our compute power is, that we have a super cloud run by Lincoln Labs, so our kind of sister national lab just up the road, but we don't have compute to compete with certainly companies you mentioned, as well as the universities. Second is that, I think that the top students right now are rethinking where they should go. So for those of you who maybe not have been on campus for a while, Course 6, or Electrical Engineering and Computer Science, actually now just expanded to include 6-4, which is a new major focused on AI. And interestingly, again, for those who have some legacy degrees here from the Institute, the department's actually considering dropping the circuit-based. Yeah, I have the same reaction. I couldn't believe that they were thinking, but students aren't signing up for that. So we're still getting some, but MIT has a way to go. I think folks like Stanford have done a much better job of organizing their faculty and going after larger scale challenges. I think we as faculty are a little too disparate and a little too siloed ourselves, and could benefit from going after larger moonshots, overused term, but moonshots within AI.
Maybe we open up the floor for questions. We have, I think, 3 minutes left.
How do we handle logistics here? How do we handle logistics for the questions? We've got...
Maybe just speak up.
Right. You have thousands of data. Here. How do you scale that? You have a detail. That's like you scale it.
When you say higher level, do you mean higher by number or?
Towards that. How do you manipulate it and make that?
I see. So we actually compute. So the industry only has about 700 molecules, but then we actually compute. So the target is 10 to the 11th. Now we're at about 10 to the 7th. And then our goal is to get to maybe 10 to the 9th. Once you get to 10 to the 9th, then it becomes accurate enough that you can rely on AI to predict the rest. So there's like I think a rule of 3%. Like any database, if you have actual data on about 3%, then you can use the deep learning to predict the rest.
So in R&D, I'll qualify. So we don't simulate in most cases. We'll do a data-driven model. So we'll train a data-driven model, then use the model to make a calculation of a property on, say, in our case, a compound. Your question's a really good one. So as I alluded, we've got really good data right now for growth inhibition and in some cases killing activity, as well as tox in human cells. We need to collect more data. We need additional assays. And unfortunately, a lot of the assays, the experiments that could give you the data are really slow or expensive. So I'll give a couple examples. So for example, most of drug discovery is focused on targets, target-based discovery. So a target is typically a protein that you want to inhibit or disrupt to have a desired biological outcome. DeepMind, advanced AlphaFold, which use protein structure to train a model that can now predict 3D protein structure with very good accuracy based on sequence alone. AlphaFold and other groups, DeepMind, are looking to see, can you now predict how a small molecule of the type we're discovering, of the type that Qichao's group is developing, how do they interact with proteins? And we just don't have good data to do that. We, for example, published a piece 2 years ago using AlphaFold to see could you use it to predict protein structure for bacterial proteins to then identify do they interact with the small molecules we identified to make good antibiotics. And we did no better than chance. Literally, if I asked you compound A versus protein B, guess. I don't even tell you what they are. You would do as well as these models. And what was interesting is a journalist in Science actually published an article that was entitled, "It's not AlphaFold's fault." Now, I don't know why they defended AlphaFold. But in short, it was that we don't have those data. The second example in the need on simulations, you'll hear a lot about digital twins coming up on the medical side. I hate the term and I hate the idea, in part because I think it's really hard to simulate complex systems. I had an hour and a half call with Google X a few years ago where they wanted to commit $100 million to simulate a bacterial cell to use it to gain. And I said, you can't do it. We have so many undefined parameters and it's such a high dimensional system, you can get anything out. Having said that, to get after data-driven models, I think could be critical for advancing our ability to come up with molecules that work. And so we need different kinds of data. I think we and others are now collecting intracellular data. Can we get after gene expression, protein expression, metabolite expression? How do you get after true tox data? You really can't kill 1 million mice. It's not nice to the mice, and that's an incredibly expensive project. So we have to think about how do you get biology in a dish. And so there's a lot of efforts now around organs on a chip, technology that can look at kind of reduced biology outside of a living organism. And I think there's a lot of hope, and MIT is leading a charge on that. But your question is really astute in that we need to get after key data to take advantage of these beautiful models that are being developed.
We got time maybe for one more question.
Wow.
With me here, so whoever you call.
Question, comment? Yeah.
Speaking of the data, obviously it's very expensive to collect it on the scale that we need with the number of compounds that we're looking at and to do screens. There's also value in doing screens and getting the data on very specific models because we don't know yet which models are best for different things, and at this point they're all different. So how do we keep the cost down but allow that proprietary information to stay with the companies that have invested, right? And yet not be so redundant, right? How do we have a field where not every single company needs to do all of that work to collect a massive data set?
So I think in the battery field, I think whoever is the first to map that big database, then it makes no sense. Well, it would make less sense for the other companies to do that. So the company that's the first to map and then invest in this database can monetize this. For example, we're trying to be the first in the battery industry to map this giant database so that we can monetize this. Because it would actually be cheaper for other companies to just pay us than for them to invest in the same amount to do this. So that's for the molecule side. But I think on the manufacturing side, I don't think there is a good solution to that because different companies will have different battery designs, different manufacturing process. So then it'll be hard to come up with one model that can be generalized across different lines. So then you will, so different companies will need to invest in their own lines. But the first one, I think just trying to be the first, and then the others will just pay for you.
Yeah, a really interesting point you raised. So I've been in systems biology for about 25 years now and interacted with all of the big pharma and biotech. And it's interesting is that they actually don't have very large data sets on the inside. When you dig in, that very rarely will companies make that big commitment that I think is needed to really get after insight, say, into indication A or this model. And many companies rely on public data sets, which I find generally not to be particularly valuable because they're disparate and with a couple exceptions. So I think the sequencing efforts and the protein structure database. And 2 points to that. One is, again, the AlphaFold effort that came out of Google DeepMind. Google DeepMind never did an experiment. They never actually did an experimental determination of a protein structure. They utilized this public data set that had been built up over many decades, largely with public monies. And so I think what we need to do within my space of biology, biotech medicine, is have increased commitment from nation states to create public databases that can be used broadly to address human health. One of my former students is now head of the DARPA Bio Division, and his vision is to create data factories. So can you actually support groups that would generate data that would allow us to generate these models that could give us insight from both the biology and chemistry to really embrace the complexity? And so I'm hopeful, but I think we also, it's not a glorious thing to do these large data bits, but we need those to get after these models.
Awesome. Perfect. Thank you so much, Jim. Thank you, Qichao. Amazing talk. Thank you so much.